Hi. All right. So, hi. Just let me get it. Right here. It's weird because it's backwards. All right. So, hi. I'm Jess. I feel like a loser doing this, but I'm the person behind the page Biotech Babe. Don't really think I'm a babe per se. It's more like a dig at food babe because everyone I think is really getting sick of her science BS. Just total quack. But yeah, I'm gonna do a DNA extraction for you. It's called an alkaline lysis. It's really simple. Just going to be using kind of like household items to do this. We'll walk you through it as I do it. So yeah. So I'm a biotech student wearing my awesome sweatshirt. I'm going into my second year of my undergrad. I love my program so far. We do a lot of really cool stuff. It's basically just playing stuff in the lab, which is awesome. All right. This is not a lab, but you want to have your hair back, you have long hair. None of the stuff that I'm using today is dangerous to handle, but just for the sake of being correct, we're going to use gloves. These are not lab kosher gloves either, but like I said, nothing here is harmful. Eye protection. Excuse me. All right, so today we're going to extract some banana DNA. Again, it's just simple. Nothing that needs laboratory facilities to dispose of anything because I don't want to do anything wrong. So what we're going to be, some of the items that we're using today to do this is palm olive. There's something in it called SDS that is going to facilitate part of this extraction. Ice cold isopropanol, rubbing alcohol, some NaCl, salt, sodium, sodium chloride. Usually you want to use deionized sodium, but we're just going to see how this works. It is just regular table salt. And then water. Usually you'd use distilled water because it doesn't have any impurities in it. But today we're just going to use sterile water. So the first thing that we're going to do is, so we want to set up our lysis buffer. So we're going to be using 20 milliliters of palm olive. And 20 grams of salt. I don't have a scale here at home, so we're just going to guesstimate. So this is the lysis buffer. So in the palm olive, the SDS, what it does is it breaks apart the nucleus by emulsifying the lipids and proteins and that make up the nuclear membrane. So it basically slices open like the cell, the nucleus of whatever you're extracting, so the plant cells, banana cells. And what that does is it allows the DNA to be taken out of the cell and be available. And then what the salt does is it protects the DNA by shielding the negative phosphate end of the DNA. I'm just going to mix that up. Right. And take our banana. I'm going to use this empty plastic bag. And before I do this part, I'm going to set this up just so I'm not making a mess. So I have just a filter, or sorry, a funnel and a filter, and just like a glass container. So we put the funnel in there, and then filter in. So now that I have my banana in the plastic bag, we're going to take our Lisa's pepper. And we're going to pour some into the plastic bag here. And then very gently, oh dear, without making a mess. So I'm going to squish this. I have to break it apart. This is the fun part. Just kind of smushing stuff. Trying to smush this without spilling it everywhere, breaking the bag. So right now, the cells are being broken open, the DNA is being released, and the salt is protecting the phosphate backbone. Okay, so now I'm going to take this awesome mixture. I take my funnel, filter, and my cup. And I'm just going to pour this right in here. All right. Now, we just wait for this to filter through because we don't really want any goop. You can already see that it's starting to filter through. Now, I'm just going to get myself a fresh pair of gloves because these ones are a little bit icky. So usually we'd use something called a conical tube to do this in, but this isn't a lab. So I'm going to just use like a regular glass jar. So it's just filtering it down. I have these just wooden sticks here, and this is what I'm going to use to actually pull out the DNA. Thank you, Elsa. Yeah, we're using bananas. I believe Facebook saves these, so after it's done, I believe you'll be able to click on it and watch the whole thing from the start if you want to do so. All right, so maybe I should just wait longer. And try to carefully pour our filtered solution into just the clear thing. Labeled it banana DNA. This mirror image camera is really messing me up. Alright, so carefully pour this because this isn't the best container to pour from. See, we have the solution in there. So now what we're going to do is we want to add the ice cold isopropanol. Same stuff you clean your cuts with out at home. And it doesn't have to be exactly precise, so I'm just going to eye this. So we'll pour the alcohol in. And you can see how there's two different liquid phases. The cloudy stuff, the DNA solution at the bottom, and then the isopropanol clear at the top. So like I said, you normally have a conical tube that I could seal, but you just want to gently invert this and then give it a second. So the DNA is going to be kind of like this fluffy stuff, this white fluffy stuff. I just wondering what I could like put it on so you can see. Alright. So, if I put my behind it, you can see the fluffy stuff in there. Kind of like sitting right along here. That is the DNA that I'm going to be pulling out. There's a nice little band of it. I wish the camera was clear. You can probably kind of see it. But there's a nice little band sitting there in the middle of the two liquid phases. All right. Again, if you're in a real lab, this is a no-no. Don't do bare hands, but I'm just at home and all this stuff is safe. Never eat anything in a lab, even if you're using food for DNA extractions. Just don't. All right, so let's pull this DNA out and see what we have. So here, actually let you watch. So you can see it's hard to kind of get out here. That is DNA. I'm going to get the whole little glob out. Try to. This is difficult. Usually there's not a lip. There we go. There we go. All right. This is banana DNA. So not a lot of people get to see DNA with their naked eye. That's a lot of DNA. That's why you're able to see it. If you looked under a microscope, you would see like a huge amount of DNA strands here. Now, there are other extraction methods where you can use very, very small amounts of your sample or like tissue to get DNA that you can actually use to do things with. This method is a really kind of like quick and dirty way to do it. It's not really ideal to extract DNA to actually do something with it afterwards. You can do similar methods and then do like a chloroform and a phenol cleanup step, which does make a huge difference. But yeah, banana DNA, pretty freaking cool. But yeah, so there's the DNA extraction I promised. I'm hoping to do another live when I reach maybe 1,000 likes, actually in the lab with actual equipment and stuff, where I'd be able to do something a little bit cooler. So yeah, if you can share, like, whatever, help the girl. But yeah, thank you for watching if you did, and have a great night. And yeah, bye. I know I'm a loser.